Okay, so with that, we're going to start by going over what I think are the most exciting developments in the field of single-cell genomics over the past year. So I have 8 areas to cover today, both computational and experimental advances. The goal is not to go into too much detail for any one method, but I hope to really summarize the intuition behind each one, why I'm excited about it, and maybe convince you to read it or give it a try. Everything on this list is something that we think you should be aware of, even if it's just in preprint form. And in fact, we've made a particular effort to prioritize highlighting preprints because many of you might not have seen them yet, but they could be really valuable for your work. So if you find these methods intriguing, we hope you'll check them out and read the paper. And for each method, I'll try to give some sort of historical perspective on what the field has done in the past and how these new methods contribute to the direction going forward, and to really highlight some of the older work that paved the way for where we are now. And lastly, we'll make a video of this talk available after the workshop. So I hope that'll be helpful to people. All right, so now I want to start with one of my favorite topics, which is the idea of recording cellular memory. It's one of the biggest challenges in our field, but it's really exciting. When we do single-cell RNA-seq, of course, we take a snapshot of cells. We understand what a cell is expressing exactly at the moment that we lyse it. But so much of biology is about history. What was the cell doing an hour ago? What was it doing yesterday? What did it do much in its past? What did it experience before it made a fate decision, or before it became something like resistant to a drug? So the question is, can we engineer cells to actually store a record, a stable record of what they used to express? This year, and there have been a couple of methods that came out that are very different and extremely creative. One is called Time Vault, and the other is called GEMINI. I'm going to go through them both briefly. They're just beautifully innovative pieces of work, and then you'll hear more about them throughout the day. So let's start with Time Vault from Fei Chen's group at the Broad. This one builds on a really beautiful piece of natural cell biology. I didn't know this, but cells contain these large hollow protein particles called vaults. They're constructed mostly from the major vault protein, or MVP. So they're like little barrels. And importantly, their interior is somewhat protected from the harsh cellular cytoplasm. So they're shielded, for example, from RNases that tend to chew up RNA. So Fei's group had this somewhat wild idea. What if we could stash mRNA from a certain time point inside of these vaults for safekeeping? And that's exactly what Time Vault does. It took some really clever engineering to get this to work, but I'm going to try to explain it to you. They need to trick a small fraction of cellular RNA to enter this vault. And the way that they do that is they fuse a vault interacting domain to a poly-A binding protein. So this poly-A binding protein grabs cellular mRNAs by their tails, and then the vault interaction domain sort of drags them into the vault. So just a random sample of your cellular transcripts will hopefully get docked and stored as cargo inside this barrel, and then the vault is closed and protects it from degradation. So when your mRNA is outside the vault, it's pretty unstable, as you guys know. But the paper shows that once it enters the vault, the half-life of mRNA it increases to more than 13 days. So you really can sort of preserve cellular history in this way. And what this means is that when you look at a group of cells, you can isolate specifically only the molecules that they expressed up to a week ago. You just add RNase. And when you do that, all of the cell's current RNA, which is unprotected by the vault, gets chewed up, but only the historical RNA gets protected. That's all that remains. And that's just a beautiful idea. So you get this sort of window into a cell's past transcriptome. What was it expressing back when those transcripts got loaded into a vault? And it opens up all kinds of questions about what a cell was exposed to in the past. How did it respond? How did two seemingly identical cells go on to make different fate decisions? You finally get a handle on the cell's history and not just its present. And they have some really cool applications of this in the paper. For example, in cancer, a very rare subset of cells can enter a persister state that lets them survive drug treatment. And these persisters can be a major source of relapse. The problem is that they're rare, and you really can only notice them after treatment once the history is typically lost. But with Time Vault, you can preserve the transcriptome before drug treatment, and then come back and ask what those surviving persister cells look like before the drug was even applied. So I'm not going to spoil the biological results. If you want to learn about this technology and what it found, you don't even have to read the paper. The lead author, Kevin, from Fei Chen's group, is presenting here at 3:20. So please stay tuned. Now, the second approach to the same problem is completely different, but equally innovative, and it's called GEMINI, and it's from the Lynn Lab at Johns Hopkins University. And the best way to explain it is to start with the idea of tree rings. Now, as many of you know, the rings inside of a tree effectively serve as timestamps of the events that occur in its life. So if you read the number and location and width of the tree rings, you can reconstruct the passage of time. You can learn what year there were droughts, what years were rainy, and so on and so forth. And as wild as that is, Gemini builds exactly that same idea inside of a living cell. So they use some really innovative computational protein design to create a structure that can self-assemble into larger subunits and keep growing bigger and bigger over time inside a cell. So because it keeps assembling and getting bigger over time, it actually functions kind of just like a tree ring does. And you can actually see here, they really do, these synthetic units really do look like tree rings inside of a cell. Because the rings grow outward, time is encoded radially. So the earliest events that you see on the innermost rings, those are the earliest events, and the later events are the ones that appear closest to the surface. But the really clever trick is that you can make different versions of these subunits in different colors. So you can lay down different colors at different times, and in that process, you can write the cell's history into physical rings that you can later read out by microscopy. So there are two really clever ways to add color. In the first, the experimenter adds a colored dye. You just add it to the dish at a chosen moment. And that sort of lays down a timestamp into the dish. So, if you add a blue dye at 5 hours, then you know that that's marking a 5-hour time point, and you can timestamp particular moments in your experiment. But the other way you can add color is driven by the cell itself. So you can engineer a reporter construct, driven by some cellular activity like NF-kappa B signaling, to produce a colored subunit, and that creates a colored signal ring. So that way the cell records its own activity into the rings right when it happens. So I know I've just scratched the surface of this. I just wanted to give you a basic intuition of these techniques and how they work. I'm not going to say more because Dingsheng Lin's lab, who developed this, is directly after me, and I'm sure you'll enjoy that talk very much. All right, so now let's switch gears to AI. It seems like that's most about what we talk about these days. Last year, I highlighted virtual cells, but this year there's been a different kind of advance. I'm pretty excited about it, and it builds off of a model actually that OpenAI introduced in 2021 called CLIP. And the problem was that most of the neural networks that OpenAI was building were either text-based or image-based, but they couldn't really connect the two. So they didn't naturally understand that the phrase, puppy, and a picture of a puppy were referencing the same idea. And they wanted to teach a model to do this, and the way they did it was really powerful, but actually quite intuitive. So they showed the computer hundreds of millions of images, each paired with a short caption from the internet. And so when an image and a caption went together, say a photo of a puppy with text like a dog or a pup, the model was nudged to place those two representations close together. But when it's a negative control, they paired the image with the wrong caption. They made sure that the computer nudged those representations further apart. And after seeing this over and over, the model starts to learn the connection on its own. It figures out that certain words and visual patterns point to the same concept. So this is really cool for text and images, but last year, Christoph Bach's group did something similar for cells. They call it Cell Whisperer. And the idea is they have a lot of RNA-seq or single-cell RNA-seq data. Those are like the pictures. But then for each data point, they also have textual metadata, and those are like captions. And so the idea behind Cell Whisperer is to bridge those two worlds into a shared space with a multimodal model, just like OpenAI did. So they train a neural network to project counts data from RNA-seq and paired text descriptions of that data together into a shared embedding space. And that links the molecular description of a cell with text using natural language. So once you've connected language and expression, you can do some things that feel almost magical. You can search a cell atlas using plain English. Instead of having to write code to cluster or subset or pull out markers, you can just ask, show me where are the CD34 positive cells in this data set, or where are the K562 cells, or where are the immune cells that are undergoing active remodeling. And it will tell you which of those patterns light up in which cells in the data set. And to be clear, it's not just searching the metadata. That wouldn't be hard at all. It's leveraging the fact that natural language and text have been mapped into a shared space, and it's possible now to be able to translate from one to another. I mean, the best analogy I can give you is like if you ask ChatGPT to show you a picture of a dog, it can translate between the text and image modalities to do this, and that's what Cell Whisperer is trying to do. The range of what it can accomplish is quite broad. On the querying side, you can ask it to find cells for you, as I mentioned before, and it can pull out relevant ones. You can also show it a molecular counts vector for a cell and ask it to generate a caption. So you can use that to annotate your cells or even just find out what makes individual cells or clusters unique. So it's both a search engine, but also sort of an analysis assistant for your atlas. And if you're interested in this, Maura Schaefer is giving a talk later this afternoon. They'll tell you the full story. And I think this points towards how a lot of us will interact with data in the future. But even more broadly, it points to a world in which both humans and computers don't treat molecular and textual data separately, but actually process and learn from it together. And that is something that I'm very, very excited about. I just want to point out also that a few groups have actually had this idea and made progress in this space. You can check out these papers, and I expect you'll see quite a bit more for multimodal language models in the future. Okay, so for our next method, let's pivot from artificial intelligence to natural intelligence, so studying the mammalian brain. Now, as many of you know, there have been huge investments into using single-cell RNA-seq to be able to discover and identify cell types and states in the brain based on neuronal transcriptomes. But a neuron is much more than just the RNAs it's expressing. It's defined by its wiring, the inputs it receives, and the outputs it sends that extend across a huge variety of synapses. And the key point here is that two neurons can look identical in their transcriptomes and still have completely different connectivity and function because their dendrites and axons can project over very, very, very long distances to completely different structures. The classic way to be able to study this is to use something like viral tracing with either the rabies virus or the Synapse virus. And people do this because viruses are very good at spreading both within and across neurons. So if you can track their spread, you can track their connectivity. But the downside of this is that mice don't like being infected with rabies or Synapse virus. It's for obviously toxic and affects the health of their cells. So the challenge is, are there new ways for us to be able to link a neuron's molecular state to its actual inputs and outputs? And the solution this year is called Synapse-seq. It was introduced from Evan McCosco's lab. It's the same one that introduced Drop-seq and Slide-seq. So this is one of their next big things. It starts a lot like lineage tracing. You give each neuron a unique random RNA barcode. So each cell kind of has its own molecular name tag. But then here's the twist. They find a way to attach that barcode to a targeting protein that physically shuttles the barcode to a specific subcellular compartment, in particular to the neuron synapses. So that targeting protein will help the barcode relocate all the way from the originating cell's nucleus, all the way down its axons or dendrites, all the way across the different brain regions to wherever the neuron projects to. So that's the trick. Instead of using a virus to spread the barcodes, which can be quite toxic, you engineer a protein system to pick them up and transport them. So here's how the transport actually works. It's a nice two-component system. The barcode RNA is engineered to carry a little structural loop, a PP7 loop. It's something that you can grab onto. And then you have a protein with a localization domain that's known to localize to a particular type of synapse. So you can get to choose where you want to bring the RNA based on what protein you choose. You can see that here in this beautiful image. There's a near perfect overlap in the location of the barcode RNA in purple and the location of the targeting protein in green. So the targeting is very, very clearly working. And so now for each cell, you can get two different types of information. You can get the neuron's transcriptomic identity, its cell type, as you can see in a standard visualization. It's high quality single cell RNA-seq data. But now you can also get the location of where each barcode RNA spreads to, which synaptic components its barcode reach, and that's summarized here in this dot plot. So you can start to ask which molecular cell types preferentially form synaptic connections, which different regions, and that's been the thing that's been so hard to get, that molecular state and wiring measured together in the same cells. But when you can get it, you can start to learn some really fascinating rules for how the brain is wired together. So again, I'm not going to spoil the biological findings here because Amanda Urke, who's a MD PhD student in Evan McCosco's lab, is going to give a talk on this at 12:20. I'm going to embarrass her, but I think it might also be the day of her PhD defense later. But just one last point on this. I think I'm excited by this method to learn neuronal connections. But this also seems like a very clever strategy to be able to just generally move RNA from one place to another inside of a cell. And I'm not sure exactly what the best applications are of this, but I'm sure Evan McCosco and his lab are thinking a lot about that. All right, so now we're going to pivot to one of my favorite topics for the next few methods, which is cellular perturbation. So we're going to start with the world of Perturb-seq, where you use CRISPR to knock down genes and then read out the transcriptional consequences in single cells. One paper that we highlighted a few years ago that really left an impact on me is the Genome-wide Perturb-seq paper from Jonathan Weissman's lab, where they knocked down essentially every expressed gene in K562 cells and measured the response to perturbation by single-cell RNA-seq. And so they generated this really extraordinary genotype to phenotype map. So it's a really powerful platform to be able to define gene function. So they did this in K562 cells, and other groups have been porting this approach to newer biological contexts. So genome-scale Perturb-seq experiments are feasible, and more than one lab has done them. But you can see the problem. The costs and investments for library prep are just staggering. So this paper actually did more than 1,000 10x lanes to be able to generate their data set in just 2 cell lines. For those of you that work in this field, you understand what that scale of investment is. And so the question for us is, can we make genome scale perturbation screens, which are incredibly powerful, something that just any lab can run, maybe even in a routine way? And I'm excited to share with you a solution that we released earlier this year. It's called VI Perturb-seq, and I'm going to give you the intuition behind how it works now, and then Alex Purdue, the lead author, will give a talk later today. So VI Perturb-seq is based on the 10x Flex technology. Many of you are familiar with this, but if you're not, it's a kit from 10x that does single-cell RNA-seq, but in a very different way. Most importantly, it does not detect RNA using reverse transcription. Instead, it uses pairs of pre-specified DNA probes that bind to every gene in the genome. So for each locus, you have a left-hand side probe and a right-hand side probe. When they bind together, the 2 probes can be ligated and detected. So it's actually a really innovative and sensitive way to be able to detect gene expression in single cells. And it was developed because by not requiring RT, it's more appropriate for fixed and FFPE tissues. But there are a lot of actually advantages to using the Flex kit, and one in particular is they recently released a new version, Flex V2. You're going to see that a lot today. And it was designed to massively increase the throughput that you can get from one single lane. And the short answer is it really works very well. The very first time we ran this assay last year, we didn't even try to push the system to a limit, but we got almost 500,000 cells in one lane of 10x, as opposed to around the 10 to 20,000 we typically see. So there's really an enormous advantage to using this approach, and we've built VI Perturb-seq to be compatible with and benefit from the 10x V2 technology. But there is a catch with using Flex for Perturb-seq because Flex only detects transcripts that you've designed probes against. And critically, the standard Flex assay does not capture your CRISPR guide RNAs. So out of the box, you have no way to be able to know which cell got which perturbation. Now, multiple groups, including ours, have been designing custom probes that you can spike into a Flex run. But if you want to do a genome-wide Flex library, you would need tens of thousands of custom probes, and that starts to get into the 6 figures of cost. So to get around this, in VI Perturb-seq, we associate each guide RNA with a custom barcode, but we designed the barcode to have a 2-part modular design that is optimized for Flex detection. So barcode 1 binds the left-hand side probe, and barcode 2 binds the right-hand side probe. And the cool thing about this is that before, if you wanted to measure 10,000 guides, you needed to order 10,000 probes. But now you just need to spike in 100 left-hand side and 100 right-hand side probes, and the combination together gets you to 10,000. So it's a really simple trick, but it enables us to scale up to very large probe libraries. So we designed a genome-wide Perturb-seq library called Guest List. It's based on Dolcetto CRISPR-I, so it has more than 57,000 guides targeting every gene in the genome. But all of those guides are attached to our optimized barcode set. The joke is we call this library Guest List is because sort of every gene in the genome is sort of invited to the party, even if only some of them are going to emerge as very important perturbations later. Now, this virus is available in Adgene. We've already had users request it if they want to run their own experiment, which, of the nature that of the type that I'm about to show you, to do it in your own lab. So what we did is we took that guest list library, we infected K562 cells, and we generated our own genome-wide Perturb-seq. But the payoff that I find to be generally striking is that, the original genome-wide Perturb-seq took, hundreds of 10x lanes. With VA Perturb-seq on 10x Flex V2, you can do a genome-wide experiment in a single afternoon. We only did 2 lanes of 10x Genomics Flex V2. It was amazing to me how easy and routine this was for us to do. It really does work. It gives beautiful data quality. We can use the data to understand how groups of genes work together to regulate the cell and drive its function and behavior. We can reconstruct the same types of molecular regulatory relationships that they found, the genome-wide Perturb-seq paper. It's really an extraordinary pioneering vision that the Regev and Weissman Lab and others put forward. We're just trying to make it cheaper and easier in a way that's more accessible for any lab. So one downside, though, is even if it is cheaper and easier to do to make these libraries, you still have to pay to sequence all of these cells. And this is where VI Perturb-seq has another advantage. Because it works with the fixed RNA kit, you can use it to sort for aberrant cellular phenotypes based on intracellular protein or RNA markers before you even run the 10x. So you don't have to sequence everybody on the guest list. You can save an enormous amount by enriching and then sequencing only the very important perturbations. So in the manuscript, we do a full genome-wide screen, but by enriching after fixation, we got beautiful results with just 20,000 cells. That really is an experiment that we think is accessible and doable for most labs, even if you don't have big sequencing budgets. So if you're interested in learning more about this, tune into Alex's talk. She'll be speaking at 2:45. All right, so I've been talking about Perturb-seq in cell lines, which is great for studying sort of core or essential biological processes. But the next frontier is to be able to do Perturb-seq in vivo or in situ directly in mammalian tissues. So this is actually the subject of our first keynote today, Xinjin from Scripps. But before we get to her talk, I also want to highlight some additional recent work in the field on spatial perturbation screens. So I want to start by highlighting us, I don't know how to pronounce SPAC-seq from the Zen group at Peking University. And the motivation behind this method is that cells don't live alone. They live in tissues, in neighborhoods, and they're surrounded by other cell types. So the question this method asks is, what happens to a perturbed cell when you put it back into a real intact environment? And so here's how it works. So first, just like a normal pooled screen, you introduce a library of perturbations into your cell's ex vivo, so in a dish. So each cell gets a guide RNA knocking out some gene. But then instead of profiling the cells in a dish, you place those perturbed cells back into an in vivo model. So they grow up inside a real tissue, and then you see what happens using spatial transcriptomics. And that's the really key enabling trick on the readout side, is they use spatial transcriptomic technologies to capture 2 things at once. One is the mRNA, so you know a cell state and where it sits in a tissue, but then you can also capture the guide RNA identity, so you know which perturbation each cell received. And once you have that, you can ask a genuinely new type of question with this technology. You can start to look for non-cell autonomous effects. So how does a perturbation in one cell not just change the cell itself, but actually changes how the cell responds to and changes the environment around it? That's the really cool thing to be able to look at here. Let me start with some striking examples. On the top plot here, I'm showing a slice of tissue where the cells are colored by their cell type that was inferred from the mRNA. So if you squint a little bit, you can see lots of yellow dots, which correspond to T cells that are sprinkled throughout the tumor. If you look really, really carefully, you can see there's a circled region here where there's actually a depletion of yellow dots. It's sort of an immune excluded region. And actually, that region, which is circled there, it's exactly where the tumor cells were perturbed with ICAM1. So you detect ICAM1 guide RNA. And you can quantify this, of course, across many ICAM1 knockout regions. And you can see that when you have tumor cells that are perturbed with ICAM1, fewer T cells are able to infiltrate and invade those tumors. And in fact, the paper was able to use the spatial transcriptomics data to work out a likely mechanism for how ICAM1 is necessary for T cell invasion, which is pretty cool. Here's a second really cool example. This time on the T cell side, when they injected perturbed T cells into mice, they found that different guide RNAs led to very different cell localization patterns. So this plot is a bit complex, but it shows that there's a different spatial distribution for T cells that receive the CD44 guide RNA compared to non-targeting controls. So based on how these cells were perturbed outside of the mouse, it affected where they could eventually go when they were given the opportunity to seed into metastatic tumors. So why is that? Why does the perturbation change their localization patterns? So to find out, the authors use the gene expression and spatial information to understand exactly what niches the knockout cells move towards and away from. And what they find is pretty cool. They found that CD44 knockout cells tended to get recruited specifically into niches that are depleted for SPP1 positive macrophages, which are known to be highly immunosuppressive. So you can see that both in the gene expression, but you can also see it by looking at the literal distances between any two types of cells. And so what you're learning here are the rules that determine how a gene controls a cell's address within a tissue, which neighborhoods it's drawn to, and which ones it steers clear of. And these are just a couple of examples of really neat types of discoveries that you can make with spatial Perturb-seq assays, but which would be really challenging and likely impossible to discover with a single cell type in a single dish. All right, so again, I want to highlight that good ideas rarely originate from just one source. There have been multiple very nice spatial Perturb-seq papers this year, including one from Rong Fan that came out yesterday and one from the Arc Institute. So they have, please do check them out if you're interested. All right, I'm going to switch gears again and move the discussion from engineered perturbations like CRISPR to natural perturbations and variations in humans, which is really what we study in human genetics. So for 2 decades, genome-wide association studies have given us thousands of variants linked to human traits and diseases. Here's a classic example, GWAS for a variety of traits that are related to human hematopoiesis and hemoglobin. With all these significant hits across the genome, this data came from the UK Biobank. But GWAS tells you this region of the genome is associated with this trait. It doesn't tell you the mechanism. And the question this year is whether single-cell perturbation data can help us close that sort of molecular mechanism gap and help us interpret GWAS data. So the idea that I want to highlight came from Jonathan Pritchard's lab, and think of it as combining 2 different types of perturbation data sets. On one side, you have natural perturbations from human genetics, and on the other side, you have engineered perturbations that come from CRISPR knockdowns in relevant cells and read out by Perturb-seq. So the idea is that by putting these 2 things together, you can match patterns between them and learn something really interesting. So here's how they actually combine the 2. It's 2 steps. The first step is to run a genetic burden test on the GWAS data. So you ask whether rare loss of function variants in a given gene are more common in people with a trait. For example, a trait like mean corpuscular hemoglobin, which is a very key measure of red blood cell function. Many humans just have, randomly knockouts in different genes, and you can see which ones phenotypically tend to decrease MCH levels on the left, and which ones tend to increase MCH levels on the right. So that's just from human genetics data. So how can you figure out how these work? And then the idea is that if you can do Perturb-seq in a related cell type, you can take the regulators that you found in GWAS, not just one at a time, but the whole set together, and you can ask what genes they seem to control in the Perturb-seq data. Those downstream genes likely represent a molecular trait, the actual program through which the genetics acts on the phenotype. So you can go from a list of associated variants to a more regulatory and mechanistic and molecular story. So what you're really doing is you're looking for patterns that match up between the Perturb-seq data and the human genetics analysis. So this is, for example, the same data that I showed you before. If you look for regulators that are in human genetics associated with a knockdown of MCH, when we look in Perturb-seq data, we can see that they're associated with a knockdown of the gene HBA1. And it works in both directions. If we look at human mutations that increase MCH, we can see an increase in HBA1 expression levels in Perturb-seq. So that leads to a prediction that the gene HBA1 is sort of a mirror or helping to explain the levels of mean corpuscular hemoglobin, this human phenotypic trait. And of course, it's a beautiful positive control because HBA1 encodes for a hemoglobin subunit. So this really simple example shows that by combining these 2 types of data, you can connect 3 things, a physical human trait, the genes that regulate it that come from GWAS, and the gene programs that they act through, and that comes from Perturb-seq. And I find that conceptually very beautiful. So I just showed you one example for one trait, but the authors have scaled this up more broadly, and I encourage you to read the paper. The key takeaway here is that both GWAS and Perturb-seq are ways of asking what happens when we perturb genes. And although they're very different data types, there are lots of cool things we can do if we can combine them together. All right, for our next method, we're going to stick with human genetics, but now in a very different context. Not different mutations between human beings, but different mutations for cells within the same human being. And it's a good reminder that not all of our cells have the same DNA. We often approximate as if they do, but it's a bad assumption. And of course, it completely breaks down in cancer. So we need methods to be able to genotype cells one at a time to understand this molecular heterogeneity. Now, in 2019, Dan Landau's group and others figured out how to measure these cellular genotypes during single-cell RNA-seq. In their classic genotyping of transcriptomes paper, you run a single-cell RNA-seq experiment, but then you amplify a little bit of your cDNA around the mutation to get the genotype. So it's a really powerful idea. It's been used quite widely in the field, for example, for mapping cellular states and genotypes in human bone marrow, but there are a few limitations. The first is that the mutation needs to be present on the 3' end of the gene. It's also hard to genotype a large number of loci in any one cell. And also, because cDNA is generated via RT, it's hard to get this method to work on FFPE samples. So it's incredibly powerful, but there's room for innovation. And a method came out this year from Caleb Leroux and Ronan Shalin and others at MSKCC and 10x Genomics. And it's called GIFT, Genotyping of Fixed Transcriptomes. And it's the basis for our t-shirts this year. So the trick is to stop relying on 3' capture entirely and instead build on 10x's flex chemistry, which I showed you before. That removes the 3' bias because you can put a probe anywhere you want in the transcript. And the clever idea in GIFT is that you design a custom pair of probes that flank the variant, but with a small gap in between where the mutation is sitting. And then a polymerase fills in the gap across that little hole, copying the sequence in, including the mutation, directly onto the probe. And so now the genotype is physically linked to the cellular barcode, and you can read out the transcript and the exact sequence at your site of interest together. It's really beautiful and quite elegant. And they demonstrate the success of this technology with a really neat experiment. They were able to get CD34 positive enriched blood samples from 35 patients with different types of blood cancer and different driver mutations, and they ran this protocol on all of them. And now, for every single cell, you're trying to learn two things at once: what state the cell is in from its transcriptome, and which mutations it's carrying. Not just from driver mutations like JAK2 or CLR, but they actually put in probes for 66 different loci in the same mutation, in the same experiment. That's the link that we've been looking for. You can ask which mutated clones are sitting in which cell states, which clones are expanding, and how genotype drives cellular phenotype, all in fixed samples. The results here are really impressive. Since it's compatible with FlexV2, you can get transcriptomes and accurate genotypes for hundreds of thousands of cells. A particularly impressive result is the ability to recover genotyping information from more than 40 different sites in one single cell. And once you can do that, you can go beyond looking at individual mutations one at a time, and you can start to do clonal and even lineage reconstruction. These co-occurring mutations really let you delineate which cells belong to the same clone and rebuild a clonal tree. And in fact, if you if you can profile the same patient over time, you can actually watch which clones expand and which shrink, for example, in response to therapy. And that's exactly the kind of longitudinal lineage resolved view of disease that's traditionally been very hard to get. So now, because GIFT is probe-based, it also plugs very naturally into other probe-based platforms. So the authors demonstrated you can also combine it with Visium HD to get genotype in a spatial context, which is kind of wild. There are so many cool things you can do with this technology. I can't even start to go through all of it. If you want to hear more, Sydney Blattman, who led this work, is speaking at noon. I'd strongly encourage you to go. And again, I want to say that good ideas are often thought of by multiple people. And I want to mention complementary work from another group, which also came out on bioRxiv. All right, so for my very last method today, I want to talk about a computational method to be able to analyze clonal data. And it came from Peter Karchenko's lab, and it's called clone-to-vec. And the name is an homage, actually, to a very famous ML algorithm called word-to-vec, which was published by Google in 2013. And the challenge was, how can you teach a computer that two words are related to each other? So like, that dog and puppy are similar, but banana is a different word. And the idea is that you can learn what a word means without actually defining it. You just watch which words tend to show up together in the same context or sentence. So if a model trains on enough text, it will learn that the words dog and puppy and cat tend to be in the same sentence or page, but banana does not. So that's the analogy. And the idea is, what if we apply that same principle to clones instead of words? And that's what clone-to-vec does. when you do lineage tracing with TCR sequencing or GIFT, or you get a lot of cells tagged by which clone they belong to. But if we want to be able to understand what that means and how the clones relate to each other, we're going to treat each clone like a word and the cell states around it like its similar context. So the method itself is 3 steps. It maps very cleanly onto the Word-to-Vec analogy. First, you do standard single-cell RNA-seq analysis, and you group cells by their similarity in gene expression space. Then you build a clone co-occurrence matrix. You record which pairs of clones tend to show up together in similar local neighborhoods or cell states. That's the equivalent of asking which words tend to co-occur in the same sentence. And then you factorize that co-occurrence matrix to learn an embedding. So now every clone is a point in space, and you've cloned clonal identity into something you can cluster and analyze directly. So what does this buy you? The first thing is you can identify groups of fate-biased clones. These are clones that preferentially give rise to the same types of cells with similar embeddings. So in their first example, the authors looked at a viral barcoding data set in the brain. And it's really challenging to analyze because this clone data set is very, very sparse, sometimes consisting of just 3 to 5 cells per clone. But using clone-to-vec, you can still understand how they relate to each other and start to understand how different clones contain both progenitors and are biased towards different progeny and fate that they give rise to. This isn't limited to engineered barcodes. It also works on natural clones, like T cells, defined by the TCR from 10x5' sequencing. When you embed those clones, they organize by functional state. In PBMCs, that's driven by things like mate and regulatory and memory T cells. In the tonsil, you see that regulatory T cells clones split into very fine-grained functional states. And then in tumors, you see that there are clones that are specifically reactive to different groups of neoantigens. So across development, healthy immunity, and cancer, this gives us a really general way to make clonal identity something we can better understand. All right, so I'll wrap up there just to bring it back, together. This year really had 2 main threads running through it. On the experimental side, a wave of clever molecular tricks for genotyping, doing genome-wide perturbation screens, recording cellular memory, reading cellular synapses. On the computational side, new ways to connect our data to language, to human genetics, and to clonal identity. So I want to thank all the speakers that are presenting today. Please go to their talks to hear their full stories. For those of you that have turned in to this workshop for 10 years, we're so grateful for your support and your excitement. I think one of the most gratifying things to me as I've been putting together the list of speakers today is the number of speakers who told me that they've watched, and they've watched this workshop themselves. In some cases, even when they were rotation students, and using this to sort of help them figure out what field they wanted to get into and what lab they eventually wanted to join. So with that, thanks very much for listening. We're not going to take questions